Thank you, everyone. So today we'll talk about a couple, I think it's new concepts and methods to do the mainly do brain behavior association or prediction analysis. So our goal, our research is we are really interested in better understanding of the normal brain development process, but as well as those abnormal ones related to different risk factors, including both environmental ones and genetic factors. However, the real goal is to do the early identification of risks so that we can intervene as early as possible, because we all know the earlier you intervene, the better tends to be the developmental outcomes. And so today I will share three perspectives and introduce a couple studies associated with each of them. So the first one is, the early brain development, early brain functional development. Gao just showed the structural development is very dynamic. So the same thing here. Early brain functional development is also the most dynamic and sensitive to risks. So the fact that the brain is so plastic and modifiable during this period makes it the optimal for early identification risks and intervention. So the second is for us to identify risks, so we have to do associated brain with the behavior outcomes. So, but brain behavior association during this period turns out to be complex and likely heterogeneous, even within the seemingly homogeneous populations. I will touch on this as we move on. And third one is, since we recognize the heterogeneity embedded in the brain behavior relationships, even within the seemingly homogeneous population, so we have to somehow embrace this heterogeneity for better prediction. So the first one, early brain functional development, plastic and modifiable. I'll go quickly, just a couple study on this, because we have, this study has been going on for decades, and for more than a decade now. We all know the different brains functional networks experience some very dynamic changes during this period of time. As you can see, a kind of summary paper here, we look at 9 different functional networks, and although the primary networks like the sensory motor, auditory, and visual ones are pretty much adult-like soon after birth, there are other high order ones like the default mode and other frontoparietal control networks. You can see they experience very dramatic changes during the first months of life, and you can see it's continued get established during the first year. And looking at this is another example. Looking at the amygdala, you can see the amygdala is first synchronized with the nearby limbic subcortical regions in units, as well as long-term synchronization with the sensory motor areas in units. But look at 1 year of age, simply after 12 months of age, this 12 months, there's huge increased level of synchronization between the limbic amygdala area and medial prefrontal, as well as the lateral prefrontal areas, which forms the kind of the emotional regulation circuitry that we typically observe in older populations. And from 1 to 2 years of age, you see a lot of pruning process and a consolidation process. So this is the quantitative picture showing the statistically significant growth during 0 to 1 and 1 to 2. So I don't think, I think, we would all agree that, this infancy period is indeed very dynamic and modified. So it's very dynamic. So, and it's also modifiable by different risk factors. So we have done, some research into looking at different risk factors. I just will highlight a couple examples. So there are, we have found alterations related to prenatal drug exposure. So this is a study we did back in 2015, looking at 3 groups of newborns with moms. So, from moms with cocaine exposure, and 50 moms with around 50 babies from moms with cocaine, and 50 babies from moms using other drugs but not cocaine, and 50 drug-free control babies from drug-free moms. And so we just the example. So this is also between the amygdala and medial prefrontal area. We see this kind of gradient decrease in terms of functional connectivity, highlighting this the cocaine-specific disruption in terms of brain's functional connectivity. And we did another second look into different drugs based on same population because moms using drugs tends to using multiple drugs. So that's why the reason we include a drug control group in the previous slide. But in this study, we actually coded all different drugs in the same linear regression model, so we can hopefully disentangle all the different effects from different drugs. And we don't have time to go through the details of each drug, but basically each drug impacts a different set of brain regions and different set of brain functions. And overall, collectively, these drugs impact, show significant impact on over about 5% of the brain functional connections. So that's the. Also, another example for the risk factor is maternal obesity, which is a big problem now also in the United States and elsewhere too. So this is a preliminary study looking also babies, newborns from moms overweight or obese compared to normal weight moms. And we also detect significant changes in the different brain functional circuits that are implicated in adult obesity literature, including kind of sensory cue processing, reward processing, cognitive control, and motor control. So I hope this established the notion that yes, indeed, infancy is a very dynamic period and modifiable by different risk factors. So we need to look into whether we can use the brain phenotypes to do predictions of later behavior outcomes. So but it's not simple. Once we take dive into this relationship, it turns out not simple. So first of all, our initial studies show there are indeed significant correlations. For example, this one shows that the first year thalamus sinus connectivity can predict 2-year working memory and also can predict 2-year moment skills. However, if you look at the correlation, they are weak or best moderate, right? So the correlation is 0.35 or 0.26, less than 10% of errors explained in this kind of association analysis. So this poses difficulty for do real prediction. But not only that, if you look at, we also look into the prediction relationship with 4-year behavior outcomes, including anxiety, inhibitor control, and IQ. And you can see there's more problem than just a weak. There are also kind of many-to-one problem. There are many functional connections that can that have correlations with one single behavior output. Like the anxiety, you have 4 or 5 connections showing significant correlation. As well as inhibitor control and working memory. So you do have this kind of, although they are significant, they tend to be weak, and they also tend to be a many-to-one problem. And on top of that, you also have heterogeneity. So this is another example that we look into different subgroups within the newborn population. Like this is the same populations I show you in the cocaine exposed, other drug exposed, and drug-free control groups. And we only detect significant relationships between the drug impacted connections and 3-month behavior outcomes only within the cocaine exposed group, not in the other 2 groups. So this speaks to the fact that, there are these kind of subgroup level heterogeneity do exist. And this is also corroborated by, 2 of our other studies. We looked into, normal typical developing children and children with ADHD. We find that there are indeed homogeneous relationships, but also there are these kind of relationships that are really, in contrast to each other. Like the typical developing children and ADHD or children with ADHD, they show actually opposite signs of relationships. And this is also true for children with autism. It's a kind of it's a parallel study looking into children with autism. We also see the same phenomena. Like we do detect homogeneous relationships across 2 groups, indicating a dimensional nature of this kind of spectrum. However, you do see these kind of areas that show contrasting brain behavior relationships, indicating that these 2 groups of children, they are using their brain differently. Not only they show different brain features, phenotype features, however, but also on relationship level, they also show those kind of categorical differences. So this gets to the question that, yeah, indeed you show like different subgroups, like separated by either clinical diagnosis or prenatal drug exposure. They show different relationships. However, do these kind of heterogeneity does this level of heterogeneity exist in a otherwise homogeneous sample, right? For example, in a typically developing infant sample, do we also see this kind of heterogeneity? That's the question. So our hypothesis is that yes, indeed, we hypothesize such kind of differences because we only look at the brain. There are only subtle differences. Only a bigger sample size or group level analysis will give you some significant differences. However, their behavior outcome are largely divergent. So why is this? So we hypothesize that even with similar brain, since different subgroups may use their brain differently, and this may lead to divergent behavior outcomes. So that's our hypothesis. So we did this study. It's the subgrouping structure of newborns with heterogeneous brain behavior relationships. And in this study, we intentionally just choose a otherwise homogeneous sample, including all full-term babies. There are no maternal psychiatric disorders included in this group, and also all of the babies are less than 20 hours stay in the NICU. So this is like a relatively homogeneous, typically developing infant sample, 81 infants. And we want to see whether we can detect heterogeneous brain behavior relationships. So this is what we did. And we use our functional postulation in units, but they are anatomically constrained by AL boundaries. So we get a refined postulation based on functional connectivity patterns, but still largely follow the AL boundary. But this is this is a very typical connectome that we see in neonatal studies, that like the visual network, sensory motor network, they're already highly synchronized already, including the anterior and posterior part of the default mode network. They're also kind of synchronized. So this is a typical, functional brain connectome observed in neonates. And then we relate this function. Each cell is a functional connection. So we relate them each connection to 4-year IQ, which is represent one of the behavior outcomes. And yes, so basically we detected both homogeneous 343, 8% of the connections that are homogeneous, like conventionally, in a conventional study, you look at the newness as a whole, as the same group, you detect these kind of connections. They are significant across the whole group. However, we also detect 334, very similar number that are heterogeneous. As you can see here, these are the connections that there are two. Apparently, there are two subgroups: the red subgroup and the blue subgroup. They actually show very contrasting brain behavior relationships. The way we did it is a data-driven. So we didn't. So for every connection, we start with 2 random lines, and then we associate each subject to one of the lines that's closest to the slope, so that and then we can do it iteratively, like k-mean clustering, update the slopes of the 2 lines until it best matches the distribution of this scatter plots. And so in this kind of data-driven way, we kind of show you that almost in a conventional whole group way of detection, you almost miss half of the connections, that there are indeed significant relationships, but not at the whole group level, but at subgroup level. So we further did this, like, since we have 334 such connections, we want to see if there are a subgroup of subjects, like they show similar subgrouping profiles across these 334 connections. And so we kind of coded each subject by their subgrouping profile across the 334 connections. And so basically 1, 2, 1, 2, 2, 1. So we have a vector for each subject, and we do a clustering based on at a subject level, and we detect yes, indeed, that shows 2 subgroups with similar within group subgrouping profiles, and lower, similarity, between the two groups, and you can see as here. So we indeed can define two subgroups of newborns with similar brain behavior relationships within the subgroup, but very different between the two subgroups. So this comes back to our hypothesis. So this one shows the brain's functional connectivity pattern, as you will expect from the two subgroups. So the subgroups S1 and S2, you can see they are very, very similar. And we will do the test. There's no significant differences across between the two groups. And this one, the second column shows the brain behavior relationships. And you immediately notice they are very different, right? So this is basically come back to our hypothesis. They with a very similar brain, and they have very different brain behavior relationships at the connection level and whole brain level. So if our hypothesis is correct, then we would expect also different brain behavior outcomes from the two subgroups, right? So yes, indeed, you see the 4-year IQ is higher for S1 than S2, while all the other different things like gestational age, postnatal age at scan, birth weight, sex, twin status, scanner, all those motion characters, they are the same across two subgroups. So the only difference is the 4-year IQ. So why S1 shows higher? If you see why S1 shows a higher IQ, right, than S2, if we observe a little bit more closely, you can see that the brain behavior relationships in S1 is actually show similar patterns as their functional connectivity pattern in neonates. You see that these kind of high areas here showing higher connectivity also tends to show positive brain behavior relationships, and these kind of negative low connectivity areas also tends to show negative brain behavior relationships. And this is not the case for S2. For S2, it actually tends to be in an opposite fashion, like the higher connectivity areas actually tends to show low, negative brain behavior relationships. And this can be, this is, if we do the quantitative, yes, indeed, if you correlate these two matrices, there are significant positive correlation for S1, but negative correlation for S2. And this is in particular more significant in this subset of networks within the limbic, frontal parietal control, and default mode networks. So why? I mean, our interpretation is that this S1 group tends to take advantage of the prenatal functional connectivity growth, right? So the higher the connectivity, the better the behavior, because they are positive relationship. And this is not the case for S2. So that's maybe one of the reasons why this S1 tends to show better IQ outcome, because their prenatal functional connectivity growth actually positively contributed to the later outcome. So that's that study. So we now I think we know there are heterogeneity, like there are different subgroups of infants that show very different brain behavior relationships. So how and this almost invalidates a lot of or most of the prediction models, right? For example, if most current either being machine learning models or SVM or those kind of predictions, you rely on this kind of significant or homogeneous relationship within the whole group to do prediction. You try to utilize all these kind of linear relationships or nonlinear relationships, but they're homogeneous, so you can do a unified prediction. However, this kind of unified approach cannot work on these kind of connections, right? So this kind of connections, without knowing the subgroup label first, you basically cannot use these kind of connections, because for one subgroup, the higher the better. For the other group, the lower the better. Without knowing the separation of the subgroups, there's no way you can use this kind of information in a conventional unified prediction approach. So what should we do, right? We know this exists and almost at an equal level, right? There are 343 homogeneous connections and there are 334 heterogeneous connections. We do want to take into these connections with significant predictive value, right? So this is our approach. So we really want to see, okay, as long as there are linear relationships, no matter it is on the whole group level or subgroup level, as long as there are outliers based on these linear relationships, they tend also to be on the outlier of the behavior spectrum, correct? So as long as they're linear, so the outliers in the brain connectivity domain should also correspond to outliers in the behavior domain, as long as there are linear relationships, whether it is homogeneous or heterogeneous like this. So this is the study we did. A sample of babies with newborn functional resting state functional connectivity data and 4-year IQ again. We defined 3 outliers. So, based on the previous slide, the first most straightforward way is to define the number of connections that this subject is an outlier compared to the whole group. The second one is the distance of the individual subject connectivity matrix to the group mean connectivity matrix. This one complements the first measure by detecting those subjects with without outlying connections. However, the overall pattern is so different from the group mean, putting them as an outlier too. So the third one is the variability of the individual functional connectivity matrix. So this also apparently combines the first two a little bit in terms of, if you have a lot of outlying connections, you should also have show more variability. So we define these 3. We give we give a label of each outlier to each subject by comparing this one subject against the group mean. Notice in this process, we didn't even utilize the behavior measure. So all the outlier definition is done based on brain data, right? So to test the concept that the outliers defined by brain, the so-called brain outliers, will also correspond to behavior outlier. So there's no training with the behavior data in this approach. So we have 2 samples. Sample 1, 175 babies with both imaging data and 4-year IQ data as the detection sample. And then we have a sample 2. We only have brain imaging data. This sample, we don't have they either dropout study or we didn't successfully do the 4-year IQ assessment at 4 years of age. So this sample 2, we have 218 babies. We only have the connectivity data, but we don't have the 4-year IQ. We as a validation sample actually, because as I just said, our derivation of the brain outlier label is not dependent on behavior outcomes. It's only based on the neonatal brain data. So we can actually using this sample 2 as an independent reference sample to derive the brain outlier labels and see if the outlier detection based on a totally independent sample can still predict 4-year IQ outliers. So this is the results. Among the 175 babies, we defined 19 4-year IQ outliers, and you can see the x-axis is the subjects, the y-axis is the outlier definition based on z-score. So the solid circles represent the true positive, like we detected the baby as a neonatal brain outlier, and they actually corresponds to 4-year IQ outlier. So this is 8 out of 19, corresponds to a sensitivity of 42%. And then we also missed 11 of the true 4-year IQ outliers, representing the empty circles. We didn't detect them as neonatal brain outliers. However, they are 4-year IQ outliers. And then we also have 6 false positives represented by the cross. And this overall gives us a sensitivity of 42%, specificity of 96%, accuracy of 90%. I mean, this, especially for the sensitivity, people, in a first look, it turns out to be low, right? We all are used to see, sensitivity of over 80% or something like that to be claimed as a good approach. However, remember, this is using neonatal data alone to predict 4-year IQ. And there's a huge gap between neonates and 4 years. There are 4 years of development. So all the different postnatal factors, including the environment, the family, the education, all those could also contribute to 4-year IQ performance. So the fact that using neonatal brain connectivity data alone can predict 42% of 4-year IQ outliers is actually pretty striking to me. So we actually tested, we did an independent validation based on the second independent sample of 218. The results are highly consistent. We actually split the sample into male sample, female sample. The results are very similar. And we do the 2 random split of the validation sample. The results are also very similar. The only degraded performance is we split our data into 2 scanners. Like we have 2 scanners for both samples. And if we use a sample 1 from scanner 1 with 181 babies, the results are still very comparable, very highly consistent. But if we use scanner 2 subsample, which is only 37 subjects, then the performance really degraded to a level that's not acceptable. So with that, this makes me think, makes us think, maybe it's the sample size issue, right? So because we, separate into male, female, different random split, you all get good performances. Only when the sample size dropped to 37, you see bad performance. So we actually tested this. We did kind of randomly select different number of subjects in a continuous way and see at which point the performance reaches the plateau, right? So actually, we look at sensitivity, specificity, and accuracy. Actually, around around 50, you see the curves are already kind of very close to the top level. So this is one of the conclusion in terms of since this method doesn't really use behavior outcome as a training, and we only 50 data are needed as a reference sample to extract the brain reference values and to detect brain outliers and achieve good performance, which we're pretty happy about it. And apparently this need to be validated in different samples. But this is kind of promising that this simple outlier-based approach can predict 4-year IQ outliers down the road. So apparently, we need large-scale data to replicate these findings for them to be more, useful. And also, we need comprehensive assessment of environmental factors to be able to really look into the heterogeneity. And also different risk factors. We need longitudinal brain and behavior monitoring to be able to do real longitudinal data to do this kind of prediction. And we need, as Angie mentioned, I didn't talk a lot about genetics, but we do need genetic and other biospecimen data like gut microbiome data, all those kind of data to add to in the equation. And this is one of the initiative, the HBCD initiative actually tries to do this. And this is our team. We are in the phase 1 of HBCD, waiting for phase 2. This is our local team to do the HBCD phase 1 study at Cedars Sinai, a big team. And this is my lab members. And thanks for my collaborators, and thank you.